Hello everyone, this is my final lecture on electron transport chain and oxidative phosphorylation, and in this lecture we're going to look at electron transport chain and oxidative phosphorylation inhibitors. We're also going to look at the question of ADP translocation, whereby we mean how ATP that is produced in the mitochondria is actually transported back into the cytoplasm. Then we're going to address something called the PO ratio, the phosphate-oxygen ratio, which is an indication of how efficiently an organism produces ATP from a given substrate. And finally, we're going to tie up a loose end, which is with the cytoplasmic NADH that is produced during glycolysis. And we're going to see how that NADH, the electrons from that NADH, is shuttled into the mitochondria to take part in the electron transport chain. But starting off with the inhibitors of oxidative phosphorylation. So many details of the ETC and oxidative phosphorylation have been learned through studying the effects of inhibitors. This has allowed scientists to switch on and switch off certain parts of the electron transport chain and see what happens, and that has allowed us to put together this whole puzzle of how the electron transport chain works. So for complexes 1, 2, and 3, there are inhibitors such as rotenone, which is actually used as a common insecticide. It's a strong inhibitor of complex 1. It is also a natural product obtained from the roots of plants, and in certain cultures it has actually been used in fishing. So amytol is a type of barbiturate, which is a sedative that inhibits complex 1. Demerol is used as a painkiller. It's also an inhibitor of complex 1. And 2-thionyl trifluoroacetone and carboxyn actually blocks complex 2. We have antimycin A, which is an antibiotic that actually blocks complex 3. And then we can look at some of the inhibitors of complex 4. Here we have cyanide and azide that are both potent inhibitors of complex 4, which explains their acute toxicity. Carbon monoxide is also toxic to complex 4 because it has an affinity for heme ions. However, cyanide has a very strong affinity for cytochrome C, whereas the toxicity for carbon monoxide basically comes from this affinity towards heme. And because there is a lot less cytochrome C in the body compared to heme, cyanide is much more toxic, so you need a much smaller dose of cyanide to be lethal compared to carbon monoxide. There are also inhibitors of ATP synthase. There are DCCD, which blocks the rotation of ATP synthase, and you'll remember from the previous lecture that if the rotation of the ATP synthase is going to be blocked, that means we won't have the conformational changes in the beta subunits of ATPase, and that means we cannot produce new ATP. There is also oligomycin, which blocks the proton flow through the F0 part of ATP synthase, and without, of course, the dissipation of the proton flow, again, ATP synthase cannot work, and we cannot have ATP production. The final class of inhibitors are called uncouplers. Now, uncouplers disrupt the coupling between the electron transport and the oxidative phosphorylation by dissipating the proton gradient. So uncouplers are hydrophobic molecules with a dissociable proton, and they shuttle back and forth across the membrane, carrying protons from the intermembrane space, where the proton concentration, of course, is high, and they carry them back to the matrix side, and so they destroy the proton gradient. So what happens during the electron transport chain, as we saw earlier, is that we have a proton gradient being formed the whole time as the electrons are transported through the ETC. What these uncouplers do is they just shuttle those protons from the high concentration intermembrane side to the matrix side of the mitochondria, and therefore they destroy this proton gradient. And instead of that potential energy going into the production of ATP through ATP synthase, the energy is actually now just released as heat. Now this is important because there are actually uncoupling proteins in mammals, and thermogenin is one such uncoupling protein, which is found in the mitochondria of brown adipose tissue. And it is actually used to generate a non-shivering thermogenesis, so a non-shivering type of heat generation. And this is the primary means of heat generation in hibernating animals and human infants. So hibernating animals use this uncoupling of the proton gradient in order to generate heat during hibernation. And it's actually also the same that human infants use who don't yet have or don't have the shivering response to cold. They will use these. They produce this thermogenin uncoupling protein in the adipose tissue, which allows for heat generation. This slide is just a combination of some of the earlier mentioned uncouplers and inhibitors. We see that there are also some chemical uncouplers here. 2,4-dinitrophenol, dicumarol and FCCP have the same function as thermogenin. In other words, they dissipate the proton gradient. Rotenone, amytol and demerol, as we said, acts on complex 1. There are the cyanide, azide and carbon monoxides that act on cytochrome C and cytochrome C oxidase, and oligomycin, which acts on ATP synthase. Okay, so there are, as I said, some of what this lecture addresses is some of the loose ends that we haven't discussed yet. One point that we have not looked at is cytoplasmic NADH, which comes a little bit later, but we've also said that in eukaryotes ATP is produced in the mitochondria. But the question now is, how do ATPs produced in the mitochondria get into the cytoplasm where it actually has to do most of its functions? So ADP must also be brought back into the mitochondria for processing back to ATP. So of course we have to have the ADP, the kind of spent ATP, which is produced throughout the cell. That has to travel back into the mitochondria to be turned into ATP again, and then once it is ATP, it has to be shuttled out of the mitochondria to go to the rest of the cell to do its functions. And these processes can't occur spontaneously because ATP and ADP are highly charged molecules. So even though the gradient for them on the one side of the membrane might be higher than the other, they can't just pass through the membranes through osmosis because they are highly charged molecules. They have a high negative charge. So processes of transport is mediated by a system called the ADP Translocase. It couples ATP exit out of the mitochondria with ADP entry into the mitochondria so that nucleotide levels actually remain constant. So it is an exchange, an antiport system, and we call this type of kinetics in this kind of transport kinetics, ping pong kinetics. So how does this work? The ATP-ADP translocase is a single nucleotide binding site, so it can only bind either an ATP or an ADP at a time. And that binding site actually alternatively faces either the matrix side or the cytosol side. So it will bind an ATP on the matrix side. That'll cause a conformational change in the protein so that it reorients itself and face the outside, the cytoplasmic side. Where the ATP will dissociate and it'll bind an ADP. So it exchanges this ATP for an ADP, which allows it to, which causes another conformational change and reorients the protein so that it faces the matrix again. So just as a very simple illustration, now the ATP-ADP translocase faces in this case, the cytoplasmic side where it can bind an ADP. It'll also take up a proton from this side, and this will cause the conformational change and allow dissociation of the ADP. And once the translocase now faces the matrix side of the mitochondria, it can take up an ATP, binds this ATP. This will cause an eversion again, so a conformational change, so that ATP now faces the cytoplasmic side where it will dissociate and the cycle can continue so the translocase can bind an ADP again. So the movement of ATP out of the matrix is also favored because the cytosol is more positively charged relative to the matrix. So there's an electrostatic attraction there. The ATP out and ADP in is thus a net movement of a negative charge out. every ATP transported out actually also, as I said, costs 1 proton in. So it uses part of the proton gradient to actually help take ADP in and allow ATP to go out. So 1 ATP synthesis costs about 3 protons. We'll talk more about that on the PO ratio side. But exporting an ATP costs another proton, which means it takes about 4 protons from the gradient to actually make and export an ATP. But we will talk about that more now. So we move now to this idea of the PO ratio. This is to answer the question, how many ATP molecules can be made per electron pair sent through the electron transport chain. So this is described by the PO, or phosphorus oxygen ratio. What is this ratio? The P is the amount of phosphate taken up. In other words, it's the inorganic phosphate that is incorporated into ATP. Remember, ATP is formed when an ADP is phosphorylated with an inorganic phosphate to form ATP, and that is this phosphate that we're talking about here that is being taken up. This phosphate is what we take into account for the PO ratio. The O, of course, is the oxygen taken up as the final electron acceptor on the electron transport chain. So the PO ratio then is the number of atoms of phosphorus incorporated as ATP per molecule of oxygen consumed. The significance of this is it allows us to compare substrate efficiency to form ATP. So different substrates will give a different PO ratio, and we also have, for the same substrate, different PO ratios between different types of organisms. So this gives us an idea of how efficient a specific organism is in forming ATP from a specific substrate. Now this PO ratio will depend on the ratio of protons transported out of the matrix per electron pair passed from NAD to oxygen, and on the number of protons that actually passes through the ATP synthase to synthesize ATP. And how many protons are needed for ATP to synthesize, for ATP synthase to synthesize ATP depend on the number of C rings in that F0 ring of ATP synthase. Now remember, we said that they differ between different organisms. Some organisms have 8 C subunits in that F0 ring of ATP synthase. Some of them have up to 15. And however many of those C subunits there are, that is the amount of protons that is needed for one complete rotation. So if it's a C8 F0 ring, then we need 8 protons for one complete rotation. If it's a C15 ring, we need 15 protons for one complete rotation. Now the significance of that is that because one complete rotation allows the 3 beta subunits of ATP synthase to step through the 3 different conformational states, it allows 3 ATPs being produced during that time. So for an organism with a C8 F0 ring, you only need 8 protons to travel back into the matrix to get one rotation to get 3 ATPs produced. But if it was a C15 ring, it means you actually need 15 protons to travel back through ATP synthase to allow one rotation to allow also just 3 ATPs being produced. So that is why these PO ratios can differ significantly. Now in older texts, you will see that there were kind of these traditional values ascribed to say that the PO ratio from electrons that originate from NADH is about 3, and from FADH it would be about 2. But we'll see that it actually is a little bit different from that. So current data suggests that the electron transport chain yields 10 protons pumped out per electron pair from NADH. So if the electrons came in as NADH at complex 1 and travels from there to complex 3 to complex 4 onto oxygen, during that chain you actually get 10 protons pumped out of the mitochondrial membrane into the intermembrane space. So If you have an ATP synthase with 8 C subunits in that F0 rotor, it means that you require 8 protons to come back into the mitochondrial matrix in order to form 3 ATPs. So you actually then need 8 protons for 3 ATPs to be produced. And remember, we just said those ATPs now need to be shuttled outside of the mitochondria as well, which means that an additional 3 protons are actually used in that exchange with ADP to transport ATPs out of the mitochondria. So we need 11 protons to flow back into, that is basically the cost in an ATP or organism with an ATP synthase with a C8 rotor. The cost is 11 protons to produce 3 ATPs plus transport them outside of the mitochondria. Now that means that if we just divide 11 by 3, it means we have 3.66 repeating, let's call it 3.7 protons that have to be used for every ATP that is produced and traveled back into the cytoplasm of the cell. So 3.7 protons flow back into the matrix per ATP being produced and transported to the cytosol, which means that for NADH, if we go back here and say that we suggest that for every electron pair that comes in as NADH, 10 protons are pumped out into the intermembrane space, it means that if 10 protons are pumped into the intermembrane space, and it takes 3.7 of those protons to actually produce an ATP and get it back into the cytosol, it means that for every NADH, for every electron pair coming in as NADH, 2.7 ATPs are being produced and are available in the cytosol. For electrons entering as succinate, in other words, electrons during the TCA cycle, which will go from succinate to fumarate, and those electrons are then passed on to FADH. About 6 protons are pumped in per electron pair to oxygen. So remember, from here the pathway is FADH2 is part of that complex 2. From there it goes to complex 3 and on to complex 4. But complex 2 does not translocate protons from the matrix side into the intermembrane space. And so you only get 6 protons pumped into the intermembrane space using this part of the pathway. And that means we get less energy from these electrons. Only about, it's still 3.7 ATPs that are of course needed to, 3.7 protons that are needed to produce an ATP and travel it back into the cytoplasm. But FADH2 only gives you 6 protons, which means we only get about 1.6 ATP for electrons entering as succinate. Okay, so then we also need to still talk about the NADH that was formed during glycolysis. Now, most NADH used in electron transport is, of course, produced in the mitochondrial matrix. We spoke a lot about the TCA cycle, where most of the NADH is produced, and that NADH is already available in the mitochondrial matrix, and it can interact directly with complex 1 of the TCA. Complex 1 of the electron transport chain, the ETC. However, we said that in reaction 6 of glycolysis, we have an NADH being produced by this enzyme called glyceraldehyde 3-phosphate dehydrogenase. And this NADH must also be oxidized to regenerate NAD+, to ensure that glycolysis can continue. And we spoke about this at that time. We said that in certain organisms in anaerobic conditions, we will have either lactic acid produced, and the NAD+ can be regenerated in that way. Other organisms, such as the yeast Saccharomyces cerevisiae, will produce ethanol through ethanolic fermentation, which is also a way of regenerating NAD+ so that glycolysis can continue. However, under aerobic conditions, most aerobic organisms will actually then pass these electrons on to the mitochondria so that they can enter the electron transport chain. However, NADH cannot itself cross the membranes of the mitochondria, and so the organism uses one of two different types of shuttle systems to turn cytoplasmic NADH into mitochondrial NADH. So eukaryotic cells have shuttle systems that harvest electrons from the cytosolic NADH and deliver them into the mitochondria without actually transporting NADH across the membrane. We call these two systems either the Glycerophosphate shuttle or the Malate-Aspartate shuttle. You can kind of think of it as a way of, instead of traveling somewhere with a large amount of cash in your pocket, you deposit it into a bank. You deposit that cash into the bank, you do your traveling, and you take the money out of the bank wherever you arrive. And this is the way that the electrons are kind of saved through these shuttles and then harvested again once they are inside the mitochondria. Okay, so first, the Glycerophosphate shuttle is slightly simpler. It stores electrons in glycerol 3-phosphate, which it then transfers onto FAD. And you have two different glycerophosphate dehydrogenases. So it's two different enzymes. They're both glycerophosphate dehydrogenases. The one is in the cytosol and the one is in the outer face of the mitochondria in a membrane. And the two of them work together to carry electrons into the mitochondrial matrix. So the mechanism works as such. NADH is produced in the cytosol through glycolysis, and that is then transferred. Those electrons are transferred onto G-hydroxyacetone phosphate, and it reduces DHAP into glycerol 3-phosphate. The glycerol 3-phosphate is oxidized by FAD plus to form dihydroxyacetone phosphate and FADH2, and those two electrons on FADH2 are passed on to coenzyme Q to form reduced coenzyme Q, and just as in the rest of the electron transport chain, reduced coenzyme Q can carry electrons onto complex 3, and the rest of the electron transport chain will continue for there. So in this way, cytosolic NADH is used to produce mitochondrial FADH and eventually reduced coenzyme Q. So the glycerophosphate shuttle couples cytosolic oxidation of NADH with mitochondrial reduction of FADH. So just an image to show that. So we start off with our NADH. This is the NADH, as I said, that is produced at step 6 of glycolysis. And through the enzyme glycerol phosphate dehydrogenase, dihydroxyacetone phosphate is turned into glycerol phosphate. And the NADH is turned into NAD+. This is in the cytoplasm side, and now this NAD+ can take part in glycolysis again. So then we have this transport system across the mitochondrial outer membrane, which allows glycerol phosphate to travel across the membrane while also allowing dihydroxyacetone phosphate to travel out. So it is once again, it's an antiport system which allows glycerol phosphate to cross the membrane to the intermembrane side, and dihydroxyacetone phosphate is exchanged and carried out to the cytoplasmic side. So once the glycerol phosphate then reaches this intermembrane side, the electrons are put onto the glycerol phosphate dehydrogenase FAD, which is part of the inner membrane, located in the inner membrane. So this then reduces glycerol phosphate. This then oxidizes glycerol phosphate into G-hydroxyacetone phosphate, which, as we said, can be translocated across the membrane again into the cytoplasmic side. So then the intermembrane glycerol phosphate dehydrogenase FAD is now reduced to FADH, and from there those electrons can be transported or can be used to reduce coenzyme Q, and the coenzyme Q will shuttle those electrons onto complex 3, and the rest of the electron transport chain will continue. The other shuttle is slightly more complicated. It's called the Malate-Aspartate Shuttle, but it does allow a greater amount of energy to be harvested. It consists of 4 protein parts: a malate dehydrogenase in the mitochondrial matrix, and another one in the intermembrane space. It also has an aspartate aminotransferase in the mitochondrial matrix and in the intermembrane space. It has an alpha-ketoglutarate malate carrier in the inner membrane and a glutamate aspartate carrier in the inner membrane. So this is what the basic structure looks like. Before we look at the mechanism, we have these antiporters, the alpha-ketoglutarate malate carrier, which allows alpha-ketoglutarate to be carried out of the mitochondrial matrix while allowing malate to be carried into the mitochondrial matrix. We have the aspartate glutamate carrier, which allows glutamate to be carried into the mitochondrial matrix and aspartate to be carried out of the mitochondrial matrix. And then we have two types of enzymes which exists on both sides of the membrane. We have a malate dehydrogenase, which exists both inside the matrix and inside the cytosol, and we have an aspartate aminotransferase, which also exists on both sides of the membranes. So then to the mechanism, and we'll come back to the image after that. First, in the cytosol, malate dehydrogenase reacts with oxaloacetate and NADH, and that produces malate and NAD+. So the two electrons from the NADH that was formed in glycolysis are attached to oxaloacetate to form malate. The alpha-ketoglutarate malate carrier now will import this malate that is produced in the first step from the cytosol into the mitochondrial matrix, while also exporting alpha-ketoglutarate from the mitochondrial matrix. Malate is then converted by mitochondrial malate dehydrogenase into oxaloacetate, during which the NAD+ is reduced to form NADH. So in this way, the electrons that existed in the cytoplasm have already been carried over into an NAD+ to now form an NADH in the mitochondrial matrix, and from there, of course, those electrons can be carried over to electron transport chain complex 1 and continue through the electron transport chain. To finish the shuttle, oxaloacetate is then transformed into aspartate, as oxaloacetate itself cannot be transported across the membrane, by the mitochondrial aspartate aminotransferase. An amino radical is added to oxaloacetate to form glutamate, which in the process is transformed into alpha-ketoglutarate by the same enzyme. And now the glutamate aspartate carrier can import glutamate from the cytosol into the matrix and export aspartate from the matrix into the cytosol. Once in the cytosol, aspartate is converted by cytosolic aspartate aminotransferase into oxaloacetate. Okay, so it should make a little bit more sense if you see the image with it as well. So let's start off with the NADH, because this is of course where the electrons are that we want to transport into the mitochondrial matrix. So oxaloacetate is reduced to malate in the cytosol by the enzyme malate dehydrogenase, and that means the electrons are carried onto the malate and NAD+ is released from this reaction, and that NAD+ is now free to carry on with glycolysis again. So malate is carried into the mitochondrial matrix by the alpha-ketoglutarate malate carrier. So it exchanges cytosolic malate for the matrix alpha-ketoglutarate, carries malate into the matrix of the mitochondria and alpha-ketoglutarate into the cytosol at the same time. So now our malate is oxidized to oxaloacetate by malate dehydrogenase. So an NAD+ in the mitochondrial matrix is used. It receives the electrons, so NADH is formed, and of course that NADH is now allowed to carry those electrons into the electron transport chain from complex 1. Now you'll see that these are both the same enzymes, and you might ask why it catalyzes the forward reaction. Here is oxaloacetate to malate, and the reaction here, it catalyzes the exact reverse. And this is simply regulated by the stoichiometry of the concentration of these products and substrates. So in the cytosolic side, there will be more oxaloacetate, so the reaction of oxaloacetate to malate is more favorable, and this allows malate to be formed in the cytosol so that it can be carried in. but in the matrix side, the concentration of the reaction of malate towards oxaloacetate is the favorable reaction, and this allows oxaloacetate to be formed and the electrons to be harvested. So oxaloacetate and glutamate is then used to produce by an enzyme called aspartate aminotransferase to produce both aspartate and alpha-ketoglutarate. And both of these can be carried out of the matrix. As we said, alpha-ketoglutarate is carried out while malate is being carried in through the alpha-ketoglutarate malate carrier, and the aspartate glutamate carrier will allow aspartate to be carried out of the matrix of the mitochondria and glutamate to be carried in. And again, the reverse reaction of what happens inside the matrix can spontaneously happen in the cytosol when alpha-ketoglutarate and aspartate is used by the same enzyme, aspartate aminotransferase, to form oxaloacetate. So in this way, cytosolic NADH is actually turned into mitochondrial matrix NADH, which means we don't lose the ability to form to translocate the maximum amount of protons across the membrane. Whereas for the Glycerophosphate shuttle, we actually bypass complex 1 of the ETC, which is of course one of the points where protons can be translocated. And so what this means is that when the glycerol-phosphate transport shuttle is used, only about 32 ATPs per glucose can be produced, whereas up to 32.4 ATPs can be produced per glucose if the Malate-Aspartate shuttle is used. So what this is a kind of generalized summary of the amount of ATP that can be produced in the entire catabolism of glycolysis. We have the entire catabolism of glucose. We have the glycolysis pathway here. We have pyruvate conversion to acetyl coenzyme A, citric acid cycle, and oxidative phosphorylation. And you will see that all of the steps here are identical in terms of how much ATP it produces, until we look at this part over here, which is 2 NADHs from glycolysis that yields 1.6 ATPs if NADH is oxidized by the glycerol phosphate shuttle, and 2.7 ATPs by the Malate-Aspartate shuttle. And the difference is there because of what I just explained. If you have cytoplasmic NADH through the glycerol phosphate shuttle, you form mitochondrial FADH, and you bypass complex 1, and so you translocate less protons. But through the Malate-Aspartate shuttle, through the Malate-Aspartate shuttle, cytoplasmic NADH is turned into mitochondrial NADH, and so you still have the maximum amount of protons being translocated. So in bacteria, because there is no mitochondria, you don't have to use the extra proton to export ATP into cytosol, which is another reason why organisms may differ in PO ratios. So then taking this together, there are 3 main factors influencing the PO ratio between different organisms. In other words, the amount of ATP that can actually be produced from the same amount of glucose, or any substrate really. The first, as we said, is the amount of C units in the ATPase. The more C units you have, the more protons you need to cross the membrane in order to complete 1 rotation, and you're still only going to make 3 ATPs in 1 rotation. The amount of the glycerol phosphate shuttle being used versus the Malate-Aspartate shuttle, because glycerol phosphate shuttle turns cytoplasmic NADH into mitochondrial FADH, versus the Malate-Aspartate shuttle, which turns cytoplasmic NADH into mitochondrial NADH. And then finally, the need to translocate ATP in eukaryotes, which of course you don't require with prokaryotes. Okay, so this then ends our set on the oxidative phosphorylation and electron transport chain. And in our next lecture, we are going to start talking about anabolic processes